The phospholipids and embedded proteins that make up plasma membranes can be both hydrophilic and hydrophobic. And the fluid mosaic model describes how all of these different components flow around the surface of the cell. So first of all, we got our phospholipid bilayer. Again, we have the surface of our cell, and that membrane has two layers of phospholipids. So when we look at phospholipids, we first are going to look at the lipid part. So a lipid has a glycerol and fatty acids. A triglycerol would have three fatty acids. Phospholipids have two, one saturated and one unsaturated, which creates this little bend, which creates a gap, which makes it more fluid. The phospho part has a phosphate connected to the glycerol, which is charged, and a variable group, which can either be charged or polar. That changes the component where all of these carbon and hydrogens are nonpolar because of electronegativity, sharing their electrons equally. There is no partial positive or negative in this region, so it's nonpolar. Whereas this region up here is going to have either charged regions or partial positives or negatives, which is going to change the way it chemically interacts with the environment. So with our phospholipid, if you took them and shook them all up, they would end up orienting where the nonpolar regions would form an interaction with each other, forming this kind of greasy, impermeable inside region, where the polar regions would orient themselves towards the aqueous environments. Again, we have hydrophilic, water-loving regions on the outside. So we have water or aqueous regions inside and outside cells. So those polar heads are going to orient themselves towards the water, and those are hydrophilic. Now, proteins are going to exist the same way. A protein may consist of hundreds of amino acids, and those variable in that tertiary structure, the R groups of the amino acids that are nonpolar are going to orient themselves towards this internal region, whereas the charged or polar R regions are going to interact and bond and form and orient themselves towards the hydrophilic region. And they're going to, so proteins are also hydrophilic and hydrophobic. When we think about the hydrophobic part, again, we're going to have those nonpolar R groups orient themselves in this region, and this hydrophobic water-fearing region is going to contain those fatty acid nonpolar tails, but also structures like cholesterols, which are going to orient themselves towards this middle region as well. So we have lots of membrane proteins. Again, the whole purpose of DNA is to make different types of proteins. So we can have junction proteins. And with certain organs, the cells need to stay anchored towards each other to carry out specific purposes. So proteins can anchor cells to one another. We can also have cell recognition proteins that have different kind of carbohydrates attached to the outside. And again, this is really important when we start thinking about blood typing, but also every single cell in our body has MHC1 proteins, which helps the killer T cells identify self from non-self. But even our antibody-mediated immune responses can be triggered through cell recognition proteins, MHC2s, put onto the surface of macrophages. We also have receptor proteins. Again, we can have receptor proteins internal to the cell, which would bind to steroid hormones, which would be non-lipid. However, we have receptor proteins embedded into the cell membranes to receive non-steroid hormones, and this would often trigger responses that we'll talk about in cell communication. A lot of times when we think about enzymatic proteins, we think about breaking down nutrients within our digestive tract. And some of those enzymes can be embedded in our digestive tract, but a lot of them are going to be free-floating enzymes that get squirted into the tube. Thinking about receptor proteins, a lot of times G protein or G-coupled receptors will trigger enzymes in a phosphorylation cascade, a lot of times starting with adenylyl cyclase. So this adenylyl cyclase can take ATP and turn it into cyclic AMP and start an entire signal transduction pathway. We also have channel proteins. We're going to think about this with passive transport and facilitated diffusion, where things are moving from high concentration towards low concentration. Again, it'll be a particular molecule associated with each protein. However, they're going to flow freely and fast straight through the channel. Carrier proteins we often associate with active transport, where we have to use ATP to move them. But again, this is going to be a little slower. It doesn't have to be active transport. We can carry substances across through passive transport as well, but it is going to be slower because there's going to be a greater interaction between the protein and the substance that's being transported. So overall, we have this entire fluid mosaic model because all of these things aren't just frozen in place. This is a very fluid environment, and each of these things can move around within the surface of the cell. They're not locked into position. So we've got phospholipids and embedded proteins, which have hydrophobic and hydrophilic regions, and all of those components will flow around the surface of the cell in the fluid mosaic model.